In this video, I'd like to tell you how the identification tests work. Usually, we consider identification test as a test which can give us the information about paternity, to confirm or to exclude the paternity. But you have to keep in mind that they are also used to analyze the biological traces. And before I will tell you how the test is performed, some theory, not much. So, as you know, our haploid genome consists of 3.2 billion bases. It's haploid genome, so in diploid we have 2 times 3.2 billion bases, and the DNA is organized in the chromosomes. And we have 22 pairs of autosomal chromosomes and 1 pair of sex chromosomes. And always, if we have the homologous pair of chromosomes, 1 chromosome is from the father and 1 chromosome is for the mother. For instance, let's consider pair number 1. We can see 2 chromosomes, so 1 was inherited from the mother and the second was inherited from the father. Okay, and this is the theory. Let's go further. On our chromosomes, in particular locations, there are sequences which are repeating, and they are called short tandem repeats, STRs as a shortcut. And STRs are composed of short sequences, short repeating sequences, which can be from 2 to 6 or 7 base pairs. And here you have example, homologous chromosomes, particular area, and in this area we have the repeated sequence of STR. It is sequence agate, and on 1 chromosome we can see that this agate sequence is repeated 4 times, and on the second chromosome, we are talking about homologous chromosomes pair, we have 6 repeats of this agate sequence. So we can consider that for this particular homologous chromosomes, the profile is 4,6. And why we are talking about these STRs? We are talking about STRs because they are used in identification tests since the number of repeats varies individually. So we differ from each other, not for the fact that they are repeats in particular area, but because the number of repeats makes us different. In diagnostics, there are 24 commonly used STR loci. So for every locus, the exact position is given. So on this slide you can see the name of the locus, the exact position, the physical position of the locus in the chromosome, the sequence which is repeating, as this column, and we also have the information how many repeats we can expect in the population. So for instance, let's analyze one particular locus, D5S818, it's here. It's located on chromosome number 5, so 5q23.2, the exact position. The sequence, which is repeating, is simply the agate sequence, and we can expect from 4 to 29 repeats of this particular sequence in this particular locus. Let's focus on one particular locus, locus D5S818. We have the man, and of course he has 2 homologous chromosomes, number 5, 1 from his father, 1 from his mother, and on one of the homologous chromosome he has 4 repeats, on the second homologous chromosome he has 6 repeats. So his DNA profile for this particular locus is 4,6. And we have the woman, and again homologous chromosomes, 1 from her father, 1 from her mother. On 1 chromosome she has 5 repeats, on the second chromosome she has 7 repeats. So her profile for this particular locus is 5,7. Now let's imagine that they have a child, and of course the child will inherit 1 chromosome from the father and 1 chromosome from the mother. So what can be the profile of the child? It can be 4,5, it can be 4,7, it can be 6, 5 or 6, 7. We have 4 possibilities. Every time one chromosome must be from the father and one chromosome must be from the mother. Now let's see how to determine the number of repeats in a given locus. We are still discussing the locus D5S818. We can use the PCR reaction and we can design the primers in the area which is common for the population. Because what makes us different is the number of repeats, but the DNA sequence apart from the repeats we have the same. So what we can do, we can design the primers, for instance, in the common area. So we can say here will be the forward, and we can design the reverse. Maybe let's take another color, and we can design, for instance, the reverse there. I only mark the position, of course, it's not the exact sequence of the primer. And having these primers, we can produce the PCR product, and based on the PCR product size, which depends on the number of repeats, we will calculate back the exact number of repeats. And now you can see it how it will work in practice. So the primers are designed in the common area, forward, reverse, and extra modification. One of the primer on this scheme, forward primer, is fluorescently labeled. So after the PCR reaction, if this locus was amplified, we will obtain 2 different PCR products. The shorter one, because we have 4 repeats here, and the longer one, because we have 6 repeats in this case. So 2 homologous chromosomes and 2 different products. Apart from this, the product will be also fluorescently labeled, because one of the primer, in this case forward, is fluorescently labeled. And now let us see how it will work in practice. We are performing electrophoresis. It's a special type of electrophoresis called capillary electrophoresis. Our PCR products are fluorescently labeled. We had 2 different products, so shorter and longer product, and they are migrating. Of course, the shorter one is migrating first, the longer is migrating slower. We have the laser which excites the fluorescent dye, and we have detector which is reading the fluorescent. And here we have the result. On the y-axis we have the fluorescent intensity, and here we can see the appearance of first product containing 4 repeats, and here we can see the second product which appears later, and it contains 6 repeats. So in fact, based on the size of PCR product, we can calculate the number of repeats back. And the size of the product is determined by comparison with the standard of the marker, which has the non-weight. So this is how it works, and let us see the real data for the particular locus. Data for the particular locus for the mother, for the father, and from the child. And mother has a profile 5, 7, father has a profile 4, 6, and in case of the child, we can see the profile 4, 7. So now we can see that one chromosome with 4 repeats was inherited from the father, and second chromosome with 7 repeats was inherited from the mother. Of course, in diagnostics, it's never like this that one locus is analyzed, but at least 16 loci are analyzed to confirm or exclude paternity. So now let's see the example of test results. And on the slide you can see the negative test result. So it means the paternity was excluded and probability of paternity is 0%. In this column we have the loci which were analyzed. They were analyzed, of course, in case of the child's DNA, and here in this column we have the number of repeats found in the child's DNA, and the same loci were analyzed in potential father's DNA, and based on comparison between child's number of repeats and potential father number of repeats, the paternity was excluded. And here we have example of positive paternity test. So you can see the probability of paternity is close to 100%. And again the same analysis, but this time the number of repeats which is found in case of the child's DNA, you can also find in case of the potential father's DNA. So every time one number is exactly the same, because one chromosome must have been provided from the father to this child. It's never 100% because it's the statistic and it's the probability calculated, but the chance that there is another man with exactly the same profile is close to zero. In the previous tests, which have been already discussed, the idea was to confirm or exclude the paternity. So to confirm if potential man is a father or to exclude that potential man is a father for the child. But it can also be the other way around, and it was the case in 2003 when they captured the man suspected to be Saddam Hussein, but the identity was not confirmed. And to provide the proof that suspected man is indeed Saddam Hussein, they analyzed the DNA of Saddam Hussein's sons. And based on this analysis, they could confirm that the captured man is indeed Saddam Hussein.